Good afternoon, everyone. My name is Stephanie Mohammed, and I am going to present my PhD seminar, the work that I have conducted so far when I looked at the effect of 150 kilohertz electromagnetic radiation on the hypothalamic pituitary ovarian axis in rats induced with polycystic ovaries. This project was funded by the School of Graduate Studies and Research Development, and this is the CRP number. The connection that regulates the reproductive system is referred to as the hypothalamic pituitary ovarian axis. This axis starts in the hypothalamus, where we have a group of secretory neurons that release gonadotropin. When they secrete this gonadotropin, passes through the hypocele portal, where it then acts upon receptors found on basophilic cells in the anterior pituitary. This then contributes to an array of intracellular mechanistic reactions, and then it releases LH and FSH. This is released into the blood where it then binds on receptors found in certain follicles in the ovary. When this happens now, we have a proper feedback mechanism occurring, and then the ovary will release a oocyte for either menstruation or reproduction. Sometimes this does not occur, and a lot of problems happen. And one of the problems that occur is known as polycystic ovarian syndrome. This is where ovulation does not occur, and small fluid-filled sacs develop inside the ovary. The follicles end up containing more theca and less granulosa cells. So what this means is that there's an array of follicular development where we have primordial to primary to secondary to preantral antral. As these follicles develop, we have supporting cells in the ovary that comes around here and forms granulosa cells to secrete FSH, and then we have the formation of theca layer cells to help form FSH, right, to release that. Now, in polycystic ovarian syndrome, the follicles contain more theca and less granulosa cells, and the LH levels become higher, which is almost 3 times that of FSH because we have more theca layer cells. Androgen levels therefore become high. Now, why exactly choose polycystic ovarian syndrome when there are so many different problems that occur in the ovary? Because 1 in 10 women are affected by PCOS. Also, 6 to 12% of reproductive age females, which means almost 5 million persons in the US, has been reported to have PCOS. And this presents with it multiple small cysts in the ovaries, irregular menstrual cycles, weight gain, infertility, hair growth, and acne. It also brings about more chronic conditions such as type 2 diabetes, hypertension, and heart failure. The current treatment options to date are diet and exercise, pharmaceuticals, and then we have something known as laparoscopy and ovarian drilling. This works within the radio frequency, and since the birth of electrophysiology, we had electrosurgery, electrocuty, diathermy. These all work within the radio frequency, where they use the application of heat upon tissues in order to come about with some type of outcome, right? Now, what also are treatment options is where they use different plant extracts, but in this seminar, we are not interested in that. We're just looking at electromagnetic radiation, right? So novel treatment options that persons are currently exploring is the use of electroacupuncture on women with PCOS. And what they did is that they observed that it normalizes the estrous cycle, restores gonadotropin secretion, and increases protein expression. Uh, High intensity ultrasound surgery on women has also been used. They found that it was feasible to induce localized thermal damage inside the ovaries. So you can see people are actually working within the radio frequency. Now, we conducted an experiment where we used the intermediate frequency, which is a little lower than the radio frequency, and we used it on rats that consisted of polycystic ovaries, and we found a reduction in the cystic follicular size and also the formation. So you might say, okay, well, why exactly do you want to use the intermediate frequency? The intermediate frequency is in the range 100 kilohertz to 300 kilohertz, and it's confirmed basically to defined as, tumor treating fields, which is TTF. This was developed in 2004, and what it is, it's that it is low intensity alternating electric currents which can induce apoptosis, which means programmed cell death. This frequency is able to inhibit tumor growth in various cancer cell lines, and it has been FDA approved for the treatment of recurrent and newly diagnosed cases of glioblastoma. Currently completed trials are all these, and if you look at here, they are also used on recurrent ovarian carcinoma. So we decided that, okay, if they are using this particular frequency for the treatment of cancerous cells, why don't we try it on a tumor that is non-cancerous, such as cystic follicles, right? And the mechanism of action so far is that it works within the cell cycle where we have dipole alignment and dielectrophoresis. So this is what they have proposed for tumor treating field, right? So from this, we decided to hypothesize that exposure to this particular frequency could reduce the number and size of cysts formed during polycystic ovary development. And the dysfunction in the HPG axis resulting in PCO is actually improved when 150 kilohertz is applied non-invasively. This study was characterized as an experimental and observational study. So with that, we had three major objectives in which we wanted to find out, which was one, to determine the effect of full body exposure to 150 kilohertz electromagnetic radiation during the development of an estradiol valvarate model. So this was confirmed in the MPhil level, which you all would have seen, but we need to reconfirm it again since it is a new experiment, right? We also needed to determine the histopathological changes of the HPG axis in PCO rats. We needed to evaluate the LH and FSH hormone receptors and also the hormonal level for LH and FSH. Um, all this that we're doing is basically looking at everything on the receptor, cellular, tissue, and hormonal level. So our final and tertiary objective is to be able to correlate all three together, right? Sorry, all four. When we did our literature review, we were able to see that, okay, at the extremely low frequencies, persons conducted experiments with electromagnetic radiation. At higher frequencies, they conducted. But there was a gap in the literature where no one looked at the intermediate frequency to see is there any effect on follicular development. So this is the work that we published in BMC journal, where it is we were able to find that that cystic size formation and the sizes were reduced, right? So now we have actually completed the gap in that literature. Um, Also, we were unable to find any literature stating the HPG axis for follicular development. So we decided to break it down and search, right? So we found the effect of electromagnetic radiation on the hypothalamus. This is when they use mobile phones and they actually saw that it may alter brain histology and it impair its function. When they use the same cell phone radiations on the pituitary gland, they saw that it also created some type of problem, some type of alteration, which could be harmful as well, right? We looked at a study on the intermediate frequency where we saw they use VDTs, those are video display terminals, and these actually are sources of intermediate electromagnetic radiation, right? And they found that there was no effect on the reproductive system, right? We also looked at it on the field exposure to human neural cells, and they said that it doesn't cause any type of stress responses. They didn't find anything serious, right? So from that now, we decided, okay, we needed to conduct a series of experiments. So these were the experiments that we conducted. This was at the MPhil level, which will have been presented before. And this is a post-irradiation that we did. So we observed that in the post-irradiation, which is when you remove the electromagnetic radiation, we were able to see that the cystic development was reoccurring, right? We also conducted an experiment for the HPG axis, and when we conducted this, we were able at the histological level to see some changes. So we decided to reconfirm it and conduct another experiment where we could use immunohistochemistry and look at it at the receptor level too. Um, the procedure was performed in accordance with ethical procedures for animal experimentation, and we use female Sprogdowley rats, that is like newly cycling rats, right? We use basic housing conditions, standardized conditions, and the animals were divided into four major groups. One are control, an estradiol valve rate, an electromagnetic radiation, and an estradiol valve rate exposed to electromagnetic radiation. So this is a regular control, and this was a positive control, right? We monitor the oestrus cycle, and then we induce polycystic ovaries, right? To confirm whether it is they had polycystic ovaries, vaginal swabs were taken, and once we saw persistent vaginal cornification, we knew that, okay, the rats them had PCO. Then we perform histology, and this is the procedure we use, and also immunohistology. This is just a short video to explain what we did. So as you can see here, this was the exposure of the 150 kilohertz electromagnetic radiation from a Kenwood oscillator, and it was shielded with aluminum foil, right, from the control. The rats were orally administered estradiol valve rate. Their body weight was taken to monitor them, and they were anesthetized and euthanized according to proper procedures. Blood, hypothalamus, pituitary, and ovary was collected, and these were sent for histology. So the samples, the HBO, were placed on slides to maintain the tissue structure, and they were stained with H and E, hematoxylin and eosin, in order to identify specific cells that we were interested in. They were analyzed with a microscope, right? The blood that we collected, it was sent for ELISA testing. It was collected in yellow vacutainer tubes. They were centrifuged using micro centrifuges, and then the serum was used for ELISA testing, right? Immunohistochemistry was performed, where it is slides were actually placed in a retrieval buffer, and then this was to be able to help release the cross-linking of proteins in order for antibodies to combine to the specific antigens, right? When this was done, these slides were actually blocked, and the blocking of these slides was very important in order to like remove extra expression to separate proteins, right? The primary antibody was added so that it will bind to a specific antigen, and then it was counter-stained with hematoxylin and eosin. When this happened, it was then read with an Olympus microscope, right, with a pathologist. And here you can see the, the affinity. Then we use scanning electron microscope, where we just dehydrated samples for the ovary, and we gold plated it using an EM spanner coater, and then it was read using a scanning electron microscope, right? Okay. The results that we obtained, the analysis was used, we used SPSS for analysis and ANOVA for, since we were dealing with four groups, to compare it, and significance or statistics was done at the 95% confidence interval. So in terms of our results, we also looked at the food intake, right? We did not find any significant difference in the food intake of each group. However, also if there's no significant difference in the food intake, then the body weight, which was for every 2 weeks we did it because it was an 8 weeks exposure, right? We did not find any significant difference. Also, we used the 7 animals for each group beside the control, because we only used 4 animals, which was just less needed, right? The next thing is that we needed to reconfirm that the EMR does reduce the size and number of cysts. So here you can see control, they had no cyst formation. The estradiol valve rate, where it is, they had plenty cysts. EMR, we did not see any cysts forming, and EV and EMR, we saw that there was a reduced amount of cysts. Some of them actually did not have any cysts, right? So there was a reduced cystic size number, so it was reconfirmed for this experiment. This is the histology of how the HPO axis look like. So this is the accurate nucleus of in the hypothalamus. Here, these nucleus contain these cells that the neurons that we were interested in. This is the pituitary, where you contain three major types of cells. So you have basophilic cells, where you will find like the purple staining nucleus. If you see pink staining, then basically those were known as acidophils, and anything with clear like these here, those are known as chromophobes. So we were interested in the purple staining one because those are known as basophils, and they are the ones that secrete LH and FSH, right? And we also looked at the ovary, the follicles, and like the cystic follicles, right? So in terms of the histological analysis, in the accurate nucleus, we form high density of neuronal cells, right? And we saw that the metabolic activity was normal. So how did we actually look at the metabolic activity? So depending on how purpley stained that these neurons are, then it means that, okay, they were metabolically active, right? So there was no difference there at all were the same. When we looked at the distribution of the basophils, we saw that with the estradiol valproate, the basophils reduction had reduced, right? And with the EV, because we are more interested in this group and this group, the electromagnetic radiation EV group, the basophil numbers was much higher. So this was surprising to find out. Then we noticed that cells were all tightly packed. There were normal shaped cells in the control and EV. However, in those in those exposed to electromagnetic radiation, we saw that the shape of the cells was irregular. We also saw many acidophilic cells in those with EMR exposure, but normal acidophilic cells in the control and the EV. Blood congestion was only observed in the estradiol valproate, but it was not observed in any of the other groups. In terms of the analysis of the ovary, this is like the primordial primary angiographian. We needed to compare them. And what we found, because this is the two major groups we were interested in, is that the estradiol valproate group had degenerated preantral and antral follicles. There was an absence of corpus luteum. So this needs to change. This is luteus, but it's supposed to be luteum. We had unilateral layer of granulosa cells, thickened theca layers, distorted zona pellucida, multiple cystic follicles, and we even had the tumor formation in one of the animals. And this was actually observed in other animals too when we conducted the experiment prior, that at least one animal had a tumor, right? Little distortion in the, so when this same group, the PCO group was exposed to electromagnetic radiation, we observed little distortion at the antral stage, presence of corpus luteum. There was Graftian follicles present. The granulosa cells were much thickened, but there was pycnosis. We had normal theca cell layers, reduced number of cysts, and the antral cavity of the cells was much smaller, right? In terms of the immunohistochemistry, this is just a visualization of how they were for each group. So wherever you see the brown, this is like it's the reactive cells to this particular so FSH, right? And here, in terms of the ovary, we looked at mainly the granulosa and theca layer cells, right? So when we were analyzing it, we used a qualitative analysis where it is, we looked at the distribution of reactive cells. And if there were no cells, it was referred to as A. If it was less than 10 cells per high power field, it was referred to as low. And then we had medium and high. We also looked at the reaction intensity of the cells, whether it was weak, medium, or strong, meaning like how strong the staining was, right? So in the hypothalamus, almost all of them was observed to be like either medium or low to medium cells, right? And the staining was, beside in the estradiol valproate, it was just a little weak, right? In the pituitary, however, we saw for the FSH reactive cells that there was high amount of cells and strong staining. The ovary, we saw that in the estradiol valproate, it was low to medium cells. But when the electromagnetic radiation was added, we saw that it was medium to high cells, so it had increased a little bit. So this is how it looked at the immunohistochemistry for LH reactive cells. And this here is the third ventricle, and this is the accurate nucleus, right? So we use the same qualitative analysis, and in the hypothalamus also we saw same type of cells, which is like low or almost absent. In the pituitary, however, there was low amount of cells with medium staining, however, reactive cells, but in the EMR group we saw that it had increased from medium to high. In the ovary too, it was low cells, but it had increased as well to low to medium cells. So this was a very important observation to note. Now, the results now for the blood parameters, the first thing is that we had two microcentrifuges, and the samples from the control group were placed in a separate microcentrifuge, and they were actually broken, and we still cleaning up after that, right? Because the blood actually stuck up in places we couldn't even clean. So we had to use the electromagnetic radiation as a positive control. When it is you use estradiol valvrate, there is supposed to be a decrease in LH and FSH. So we did observe that there was a smaller value, but in terms of the ratio, you could see that there was a higher LH to FSH ratio, right? Now, when electromagnetic radiation was added to an estradiol valproate group, we observed that there was an increase. It was not a significant increase, but there was an increase in the blood parameter for the LH and the FSH. So this was important to note, right? In terms of analyzing it now at the HPO axis, the hypothalamus, we observed that there were no changes in the density of the neurons and the metabolic activity of the neurons found in the arcuate nucleus of the hypothalamus. We did not observe any significant difference in the distribution of LH and FSH reactive cells in the hypothalamus, right, among all the groups. In terms of the pituitary now, we observed that the number of basophils increased significantly in the estradiol valproate and EMR group. There was a high distribution of FSH reactive cells among all the groups in the pituitary, and there was an increase in the distribution of LH reactive cells in the estradiol valproate group exposed to electromagnetic radiation. In the ovary, which was reported prior as well to in a publication, there was a decrease in the size and number of cysts formed in the estradiol valvrate and EMR group, an increase in the secondary graphian and corpus luteum in the estradiol valvrate and EMR group. We also saw an increase in the LH reactive cells and an increase in the FSH reactive cells in the ovary. So this was really good. Now, the intermediate frequency in general is considered to be falls within the non-ionizing category, right, on the electromagnetic spectrum. And basically, they say that it is supposed to only generate heat, but not have enough energy for the release of an electron from an atom. So we needed to kind of understand what exactly is happening. So if you look at laparoscopy, where they use a localized radio frequency, in general, this is how the polycystic ovary will be, right? So you have like dysfunction of angiogenesis, increased granulocytic layer cells. You have the cortex thickening here, intraovarian fluid increasing as well. But laparoscopy, when the application of heat is applied to the tissue, you see like the cortex, the cortical layers are loosened. You have like removal of intraovarian follicular fluid. Stroma cells are destroyed. So the heat, this is what it is doing from the radio frequency. So in general, you could see that like deep heating causes like vascular and metabolic changes. Superficial heating is good for like general menstrual pain. And then local heat could reduce like muscle tension, relax abdominal muscles to reduce pain. But heat also could like increase pelvic blood circulation and diminish congestion and swelling. Now, the type of exposure that we did was a full body exposure. So we are uncertain like what exactly the heat that is being generated and what it is doing, which is very, very important because this heat is not supposed to cause any type of ionization, right? So we're not going to give every single thing out because we are still, we have a few hypotheses, but we don't want to explain everything for this seminar, right? Now, limitations to this study was that heat was not measured in the tissues. We did conduct an experiment on male rats, and that has been sent for publication in the in Bioelectromagnetics journal, and it did come back. We made the reviews, and it was sent back. So, but when we looked at the rectal temperature of the rats, then we did not see any changes in temperature. So we know definitely that was not happening. In terms of these sections for the ovary and the hypothalamus, we use only 5 sections to quantify. So when we say 5 sections, like, it means that when we hold the entire ovary, basically we will cut here first, well, dissect, sorry, and then we will count to 3 other sections and then dissect again. And that's what we did, right? So I feel if you actually use every single thing, then you could quantify it better. And ultrastructure evaluation was not performed. We use scanning electron microscope, not a transmission microscope. So this is how the pictures from the scanning electron microscope were looking like. So this is the ovary of a normal rat. You can see like the cysts here, not cysts, sorry, corpus luteum. You can see follicular development different sizes. This is how here an ovarian cyst, an external ovarian cyst looks like. Fibrin matrix, and then you see the cuboidal shape of surface epithelium in the group that was exposed to electromagnetic radiation, the EV group, right? So it was not like clear enough for us to make like a proper or a confirmative conclusion from this, right? So this was one of our major limitations. In terms of our final analysis, at the receptor level, we believe that there is an increase in LH reactive cells in the ovary, increase in LH reactive cells in the pituitary, and no effect on neuronal cells in the hypothalamus. At the cellular level, the number and size of cysts in the ovary is reduced. There's an improvement in the number of basophils in the pituitary and no change in the neurons, right? At the structural level for like tissue, we observe that the shape of the primary and secondary follicles, like up, it did cause some type of structural change on good follicles, but on bad follicles, such as cystic follicles, we did not observe anything. Appears to cause irregular shape of cells in the pituitary and no effect on the neurons in the accurate nucleus. And in terms of the biochemical parameter, we believe that there was an increase in the level of LH and FSH, which would have been seen from our analysis. In conclusion, we could say that the effect of this particular radiation on rats induced with polycystic ovary has no effect at all on hypothalamic neurons, increases the number of basophils in the anterior pituitary, and reduces the number of cystic follicle formation in the ovary. Simultaneously, we could say that there's an increase in the number of LH and FSH reactive receptors from both the cells in the pituitary and follicles in the ovary. For this particular experiment, I would like to thank my supervisor, the main one, Dr. Zuzikov, and my co-supervisor, Dr. Sundaram. And we wish to thank our advisory committee members. And then we also want to give special thanks to Dr. Janelle Johnson. She was able to provide us with all the rats that we needed. This is Mr. Lestag. He's a technician who helped us throughout every single thing. This is Mr. Gerard. He was in charge of all the histology. Mr. David, he did the scanning electron microscope. Mr. Nicholas, he was the person who helped me with ELISA testing. Mr. Ryan, he was the person who did the design for the HPG axis. So the same background picture that you're seeing, he was the person who did this. This is Miss Maggie. She was the person who performed the immunohistochemistry, and she performed it under Professor Rao. And Professor Rao is the person who did the reading of the immunohistochemistry slides for me. So I wish to thank everyone for being here today, and I appreciate everyone that are here. Thank you all very much.